please welcome Craig Shimasaki. Thank you.
Well, somehow I got to be the last speaker yesterday, and I'm the last speaker today. The only advantage I can think of that is I can probably ignore the timer, and I might be able to get away with it. But I do want to thank all of you parents for being here, because it's because of you that we do what we do, and it is the force that you provide, giving hope to your children, to yourselves, but also to encourage the frontline physicians who do the work, and certainly the researchers. So we thank you, and it's because of you that many of these things are going to proceed forward. So what I'd like to share with you is the autoimmune antibodies and the biological basis of that. There's three things that I would like to cover today, and one of them is just these biological models about autoimmune encephalopathies, and that PANDAS and PANS is just one of those, but it is also a model for other autoimmune neurologic disorders. And then briefly about the role of what we call molecular mimicry, and how not only does it impact PANDAS and PANS, but many other chronic autoimmune disorders. And then talk briefly about some of the common infectious triggers. And then just share with you some recent data that we have actually in a manuscript that's being reviewed. So also I would ask also the slides that are in that study be held until the paper actually gets published. But some very interesting things that are developing in this area, and I think the underlying pathophysiology, the biology, the clinical presentation, the ability to diagnose and treat is going to change this for everyone, but also I think it'll make it easier for all of these other disorders. So briefly, as you know, the immune system here is actually interacting with the central nervous system in various ways. As you saw Dr. Platt talk about how the T cells actually get into the brain and then actually, like a Trojan horse, open the gates and allow the B cells to come in from the blood, which is one very good biological mechanism. And as we know that the normal immune response mounts different types, both from the innate and the innate adaptive immune system. So we've got one very strong system in our body that is directed against ourselves. So as what's been mentioned previously, that this is not a new syndrome. Actually, back in the 1800s, Sir William Osler described this percivative behaviors in children. Dr. Pasternak talked about even where Thomas Sydenham back in the 1600s had described this even in papers buried in the literature. So sometimes if we just kind of look at history and we review history, we'll find many of these things are not new. You think about really the advent of hand washing and the sudden stoppages of deaths when in the delivery room many centuries ago, before the germ theory of disease was understood. Biology and science has to evolve. So Sydenham, Chorea is the medical model for PANDAS and PANS, and it was also called the St. Vincent dance, as was described yesterday, but it involved loss of motor control, loss of emotional control, but it started also with a group A streptococcal infection. And the infection actually then induced rheumatic fever, and rheumatic fever is actually where these antibodies began attacking the heart valve. The treatment for that is antibiotics, and also these patients that develop Sydenham or that have rheumatic fever are susceptible to Sydenham chorea because those antibodies or other antibodies recognize a part of the brain called the basal ganglia. So that's how it ties into PANDAS and PANS. But if we look at the nomenclature, there's a whole lot of acronyms, and as Dr. Latimer described, that the term PANDAS does need to be modified or adjusted, but we can thank Dr. Sweeto for identifying these and defining it in a way that can begin the actual study and the clinical following of these patients. So you see PITANS and now PANS, which eliminates the S for strep, but it still keeps the P for pediatric. And as many of you know, that this is not just limited to pediatrics. So what are the common denominators? There's also PANS, CANS, PITANS, PANDAS, all kinds, but there even is, I think Dr. O'Connor described dire, so we may have another term, but infectious autoimmune encephalopathy, infectious autoimmune encephalitis, post-infectious autoimmune disorders of the brain, or post-infectious autoimmune disorders of the basal ganglia. What do they have in common, actually, all these names have in common? They're all infection-triggered or environmental. Something has to occur. They are also autoimmune-based, meaning this is an immune-triggered disorder. They're neuropsychiatric in the sense that they actually trigger the immune system to disrupt the neurologic system, but they're also directed against portions of the brain that control many of these functions that we've described. One of the other criterias for PANDAS and PANS is this acute onset. And acute meaning that your child was one way one day and at 5:03 the next day they're no longer your child. And that is important because anytime you study a disorder, you need a homogeneous population. And in order to identify biomarkers or clinical syndromes, you have to have hopefully a homogeneous population so you can make conclusions. But unfortunately, what it does is it eliminates for treatment, or at least makes it more difficult, for those that don't have the sudden onset. They may have this subacute, subclinical, or maybe what we call gradual onset. But I would argue that gradual onset must have at least some beginning of an onset, that in order to make a term gradual, because you have to know that it started, it just may not be the complete constellation of all the symptoms that are required for a diagnosis of PANDAS and PANS. So in thinking about this, I'd like you to think about this axis of this interaction between infectious triggers or environmental triggers of all forms, and then the immune system. As we've talked about, you see the T cells, the B cells, the microglia, activation and all types of inflammation. But then it attacks and ends up affecting the brain. But that's not the end of the story, because it all happens in the context of a genetic predisposition or an immune system dysfunction or susceptibility to this particular type of disorder. And not necessarily do they have a family history of neuropsychiatric disorders, but there generally is some family history of an autoimmune dysfunction, which is really just the inability to faithfully recognize self from non-self. And as that's done and as that's interrupted, the ability then to determine when something is self and non-self and make targets against it sometimes goes awry. So I want to use an example for you, I think, because the question is, why doesn't everybody get strep? 90-something% of people who get strep, why don't they all get PANDAS? Well, I'm going to use the example of lung cancer. Greater than 90% of smokers get lung cancer. I think it's somewhere maybe 95, 98%. But we also know that if you stop smoking, we can really virtually reduce lung cancer to very insignificant levels. But what most people don't realize and understand, that only about 15% of smokers who smoke all their life end up getting lung cancer. So you kind of scratch your heads and you say, okay, why do only some people who smoke get lung cancer? I can change that to say, why do only some people who get strep get PANDAS? And it's the same corollary, is in lung cancer, the difference is in the genes that control cell cycle, DNA repair, carcinogen metabolism, and other types of genetic functions, which again is an immunological dysfunction, go awry. And so you get enough of the insult over time that these patients become susceptible to these things that we call autoimmune neuropsychiatric disorders. So I hope that explains. I think this is the conundrum that makes it difficult to understand in the scientific and the medical community why there's not a straightforward association, but there's more to the story than just getting strep. I want to change the subject here for the role of molecular mimicry in triggering these disorders, because it's in and found in the case of many other autoimmune diseases and autoimmune disorders that are also chronic. So if you look at Guillain-Barré syndrome, Campylobacter jejuni tends to be the one that is most commonly associated with the onset of Guillain-Barré. Influenza is also, but the commonality that's been seen is this carbohydrate at the terminal end, which is also identical to the carbohydrate or the ganglioside GM1 on the myelin sheaths. And so what we see then is this commonality in the context of a patient who has an immune dysregulation or the potential for immune dysregulation ends up making or allowing autoimmune antibodies to circulate that attack the myelin sheath, which makes it a chronic condition that causes these demyelating syndromes. So if you look at the effect of strep, and Dr. Pasternak done a wonderful job talking about the numbers of different variants of M proteins in the group A carbohydrate and strep, I think he said over 260 or so different varieties, that the prevalence of not only new strains, but the number of genetic differences. Well, these are also associated with basal ganglia, the things that attack the basal ganglia, also systemic effects, the heart valve. Obviously, what we talked about was rheumatic fever, joints and kidney. But we also see other infections, as I mentioned, Campylobacter. You can see Epstein-Barr in lupus. You can see EBV, measles, and HHV6 in MS. You can see HSV, and the list goes on. And this is a paper by Cusick who identified that there are very common triggers. They may not be the only trigger, because if you think about it, we have, 20 amino acids. If you take 20 amino acids and you arrange them, at some point you're going to have a commonality in a bacteria and commonality in parts of your body. And it's not only the commonality of the primary structure, but the secondary, which is the two-dimensional structure, but also the tertiary structure, which is a three-dimensional structure. And so there are multiple ways in which molecular mimicry can occur. So in sort of a simplified version, we all get microbial, viral, fungal infections. We all make antibodies against these infections. And they're there to protect us because these antibodies provide attack against foreign invaders. But in these patients, and in PANDAS and PANS in children, they actually, through molecular mimicry, produce these antibodies that cross-react. And as we heard from Dr. Platt, that the T cell actually allows the opening of the blood-brain barrier, and then these autoimmune neuropsychiatric antibodies cross the blood-brain barrier and influence brain function. We call that friendly fire. Again, I'm not sure where that term came from if you look at friendly fire, but it's the best analogy I can think of. Again, it's also in the context of major histocompatibility and changes in the genetics. But when we think of wartime, we think of one of the most powerful forces, our Army and our military and our Navy and our Air Force and Marines and all the forces that we have. But sometimes they get misdirected and they actually begin attacking ourselves. And in this case, we call this friendly fire. But the target of the friendly fire in this case becomes the basal ganglia. And as mentioned previously, that the basal ganglia is responsible for motor movement, procedural learning, cognitive functions, emotional functions, eye movement, and two of the other disorders of the basal ganglia are movement disorders like Parkinson's and Huntington's. Of course, Huntington's is known to be a genetic disorder, but Parkinson's is also now identified as an autoimmune attack against the alpha-synuclein. So one of the things that was talked about by Alex in Susan's talk earlier today was that they donated her brain to the NIMH, and that they had indicated that there was inflammation in the caudate and the putamen. This was a study that was done that identified, and it's very hard to do MRI studies just on one patient, but if you look at a lot of patients, what you can see is that there is inflammation in these patients with OCD, strep-associated OCD and tics, compared to healthy children. If I can hit the right button. And the average size of it increases in the caudate, the putamen, the globus pallidus, but in this case not the thalamus, which is not in the basal ganglia. And so if you do enough studies with enough compared to controls, you can identify and see that there is an inflammatory process that's going on in the brain. So there are some infectious triggers that I want to share with you. You've heard many of them today. Obviously, group A strep tends to be the one that seems to be most common. Influenza A, varicella was mentioned, EBV, CMV, Mycoplasma, Lyme, Babesia, Bartonella, Coxsackie virus. But the important thing to note is that most of these patients really and usually typically have more than one infection. So sometimes when one infection is treated and the patient improves somewhat but doesn't completely improve, oftentimes there's other infections that have not been identified, which makes sense because if you think if this is an immune dysfunctional patient, that their susceptibility to diseases are much higher than those with a normal functioning immune system. So this is kind of a sobering bunch of text. Actually, I'd like you to read those. We have done a survey on over 1,000 patients with PANDAS and PANS, and many of you, I think, are even in this room, so thank you for responding. Our goal was to be able to put together and publish data that showed not only the economic but the societal impact of this disorder on parents and children. And the question is not even, it's on here, but the results aren't on here. These are part of the write-ins. So there was literally hundreds of write-ins, which, you had to circle one of the A, B, or C, or D. Well, there are all kinds of write-ins. But I'm sure you're all very familiar with these comments here. And it is the reason that we do need change in our legislation for insurance reimbursement, and we appreciate all those that are working hard to do that. But PANDAS and PANS may really only be the tip of the iceberg because, again, remember, PANDAS refers to pediatric and acute onset. But parts of autism, ADD, ADHD, anxiety disorders, Tourette's, epilepsy, and again, not all of that, but the way that these are actually identified are a cluster of clinical symptoms. So there's not an etiological identification, it's actually the cluster of symptoms, and often it's a rule-out disorder because you're looking for other disorders that might be more common or more likely. So I'm going to show some data to you and then talk to you a little bit about the biology of these autoimmune anti-neuronal antibodies and the cell stimulation assay. And then I want to close with some summary. These are the 5 components, and we've named this the Cunningham panel after Dr. Madeline Cunningham, who performed about 20 years of research in conjunction with Dr. Swedo and many others. But this was the result of the targets and the things that she's identified and published. And they include targets of the dopamine D1 and D2, which are antibodies directed against these two receptors in the brain. Lysoganglioside, which is a surface molecule on the central nervous system. Antitubulin, which is an internal protein, and the internal protein is really a marker of autoimmunity because we see it in Hashimoto's thyroiditis, we see it in other types of autoimmune disorders, because you would not expect to actually see autoimmune antibodies directed against internal proteins in a healthy individual. And then finally, the calmodulin CAM kinase activity assay. And this is one of our most complex assays. We actually grow human neuronal cells in culture, and then we incubate serum on top of these cells. And then after they've had a chance to bind or do what they do, we wash them off, and then we pulse chase them with radioisotope P32. And then we scrape them off, collect them, and then run through the assay and count them in a scintillation counter. We compare them to controls, and we look at, has the serum autoantibodies stimulated this calmodulin CAM kinase enzyme to be upregulated above normal levels? So why is that important? CAM kinase is actually the enzyme that's in the pathway that actually upregulates the synthesis of three neurotransmitters. Those are dopamine, epinephrine, and norepinephrine. These are the neurotransmitters that are involved in transmitting neurologic activity in the brain. So an antibody that actually could trigger the upregulation of these neurotransmitters could be problematic because your body is not normally calling for it, but it's actually being produced by an exogenous antibody. The studies and the research on the protocol has been extremely helpful and important in helping to identify at least guidelines for the beginnings of the treatment of PANDAS and PANS patients. They fall into the anti-infectives, the immune modulators, and the anti-inflammatory agents. And as you heard from Dr. O'Connor today, there's some very good data that's now being generated in support of the success of some IVIG usage. So as Dr. Cunningham has shown, that the cerebral spinal fluid will actually have antibodies that bind to the caudate and the putamen, and they're actually past the blood-brain barrier. And in PANDAS and PANS patients, and Sydenham, Chorea, where you see the purple staining, indicates that they have antibodies that have bound to it, whereas in controls you don't see that. Same thing in monoclonal antibodies from SC, which is Sydenham, Chorea, and anti-ganglioside monoclonal antibodies. So as I mentioned, two of the markers that we look for autoantibodies against are dopamine D1 and D2 receptors. The interesting thing about that is that these antibodies can function in a number of ways. In some cases, these antibodies can actually bind to the active site of the receptor, or they can actually block the active site of the receptor. And what we call those are agonist or antagonist. Agonists actually act like dopamine. So the antibody can act on the active site and stimulate the active site as if it was dopamine, or it can block the active site as an antagonist and actually inhibit the transmission of dopamine neurotransmitter function. The same way with dopamine D1 and D2. So we're getting ready to publish the information where patients were positive for autoantibodies against dopamine D1 or D2. We see certain types of symptoms typically have been correlated with these. Now again, these aren't diagnostic. These are correlations that we see with these particular antibodies with human dopamine D1 and D2. If we look at the lysoganglioside, again, I'd mentioned that it is attached around the sheath around the neurons, and antibodies directed against the lysoganglioside GM1, we see these particular symptoms that seem to be correlated with these autoantibodies in patients with these levels. Antitubulin, as I mentioned, we see these symptoms association. Tubulin used to be thought of as sort of a housekeeping protein that just kept structural function inside the cells. But as you see in this study here where there were mutations in the tubulin, you see a whole host of neurologic disorders that are resulting from that. Autoantibodies that stimulate the calmodulin CAM kinase, as I mentioned, these antibodies can bind to neurons. They can stimulate the calmodulin CAM kinase 2 enzyme, which sets in motion the pathway of tyrosine hydroxylase and then the synthesis of dopamine, while other antibodies can actually block the dopamine D1 and the D2 receptors. So you've got this very chaotic autoantibodies that are impacting proper and normal neurologic function. These are the symptoms that we do see associated with CAM kinase positive. One of the interesting things that we see also is that when CAM kinase is a positive, what we do find that it's more often than not associated with an active or recurrent infection. And so it tends to let us know, again, it's not diagnostic for an infection, but we do see, as Dr. Elena Frid mentioned this morning, that we do see it in cases where there is an active infection that hasn't been identified or a recurrent infection. But again, it's also the result of antibodies that are stimulating this. And so I want to share a couple of case studies. These we have now hundreds of them. We've tested now over 9,000 patients. Many of them we have before and after treatment. This is a 24-year-old young man who presented with OCD, decreased appetite, lost 30 pounds. And again, that's not anorexia in this case. It's an OCD of food contamination fears. And you can see there was, I think, one, I can't see that far, but I think there was one positive here. The patient was treated with an immune modulator and plasmapheresis, which resulted in complete symptom reduction. We tested the patient afterwards. The antibodies all resolved to baseline. This 9-year-old young girl who had OCD, verbal tics, and stimming behavior, which is characteristic of what people might think of as autism, couldn't concentrate, sleep disturbances, urinary incontinence, dysgraphia, as you saw examples from Dr. Kovacevic today. This patient had a couple of antibodies positive, and then interestingly, this patient was caught early, and I think this is the most important thing to note. When they're caught and diagnosed early, it's found to have an infection. The infection was treated with azithromycin. All her symptoms resolved, tested her afterwards, and everything was back to baseline. And this is the importance, I think, of early detection and early treatment. Now, it doesn't mean that people who have not been detected early don't improve and don't get well. We have great stories of Garrett and others about that. But sometimes it does take longer, and sometimes it's a little bit more complicated, because if you think of the immune system, the memory B cells are becoming stronger and stronger over time as they're exposed to infection. And just like vaccines, we give vaccines to produce memory B cells so that when an infection actually occurs, the immune system is already primed, the B cells already have memory, they've already been made, and they're ready to go ahead and produce more antibodies and attack whatever the invader is. I'll just give you two more here in this case study. This is a 9-year-old girl. She had unknown origin of neuropsychiatric disorders, but she was positive later for Lyme. One of the stories is she said, "Mom, something's wrong with my brain." We're working on a case study to actually get that submitted and published. And in her, she had one or more antibodies positive. In this case, she was treated for the Lyme, and as you may know, Lyme sometimes can be difficult to treat. Many antibiotics, 3 IVIG treatments, complete remission of symptoms, and all of her antibodies went back to baseline. Another 9-year-old young man presented with 30 days post-strep with OCD tics, separation anxiety, the typical PANDAS criteria, you see positive autoantibodies, and in this case, the patient had one IVIG treatment after 1 month of diagnosis with complete symptom elimination. So again, these are cases. What I want to share with you is this study that is under review in a manuscript. We looked at examining the correlation between treatment, also patient symptoms, and the presence of these autoimmune antibodies, and to see if they were actually correlative. We identified patients who improved after treatment. This wasn't a treatment study, it was a retrospective analysis. We found 34 of them improved. We also had the panels. As you can see, all of them had at least one positive. They were all also very sick. After treatment, in the patients who improved or completely resolved, we had retest, and as you can see, the difference in their autoantibody presence. And if you look at the number of tests in the blue are the ones that are positive. Those that actually regressed or actually with completely eliminated symptoms, almost all of them had, well, all of them had a reduction in antibodies positive, but many of them had complete baseline responses. So they're a group that did not improve but got worse. As you can see, most of them were positive, but there were a handful that weren't. So, you know, what the interesting was, are these maybe false negatives, or is it that they just didn't have time to make the antibodies? So after they were treated for whatever the treatment was, we tested them again. As you can see, many of them, their antibodies began to show up. Because this is actually a metabolic test, and if you think about it, like blood pressure or cholesterol, the time in which you take it is a snapshot in time, and it's the presence of those antibodies at that moment in time, and it's the identification of which ones in this case. And as you can see, all of them actually became positive or got worse, and this correlated with the symptoms. And there was no statistical difference between the group 1 or 2 in the age or gender, no statistical difference between the time between the first and the second test. The only differences were the treatment and whether or not the treatment worked in those patients. So the sensitivity and specificity for this, what we calculated was 88% and 83% with an overall accuracy of 86%. You can see the separation between those that improved, those that did not improve, and this is called a receiver-operator characteristic curve. The more gray you see, the better it is at predicting or correlating with, in this case, symptoms. We also are running an algorithm looking at logistic regression of compiling all 5 assays into one score, and in doing that, sometimes multiple tests, no one test by itself is a good predictor of efficacy, and so that's the reason we're looking at this. You can see an improvement in this case, 88 and 92% in sensitivity and specificity. The accuracy increases to 90%. You see better separation and a better receiver-operator curve. We also did some reproducibility testing of 344 individual repeated tests, looking at the correlation between first test, second test in CAM, and then also the ELISA assays. The reason we did this is because there is a Swedish study that was published a couple years back. Unfortunately, in that study, they used the incorrect tubes that did have excipients that have been identified as interfering with the assays. And in that case, they did print a corrigendum indicating that this was done. But what we also have had to do is to show or demonstrate that the test itself is reliable, but they also, I think, in the controls, there were some differences in inclusion criteria. And so one of the things we're doing going forward is looking at prospective studies and new controls. I'm going to skip this, but just to know if you want to look it up, because I think I'm getting an indication that time is running out, but I might take a liberty to add a little longer. Autism, Dr. Richard Fry did a study here, and I'm just going to jump to the results. Autism, again, is a clinical diagnosis, but many patients that might have a clinical diagnosis of autism, as many of our patients have, often do lose the diagnosis of autism after they've been properly diagnosed and treated. And in this case, they were tested with the panel and also all the other autoantibody panels. But Dr. Fry identified about 73% of those, or actually 60% of those were positive in that cohort of autism, and 73% received IVIG. And so what he found was in all of those patients, they had an improvement in their social responsiveness scores, their aberrant behavior scores. This is a published paper that's out there, and he showed the correlation between the different autoantibody targets and the improvement in the symptoms. And you can see that and read that. But it does say that autism, potentially, a subset of them, may be due to an autoimmune encephalopathy. We have one case study with a patient with schizophrenia from Cincinnati Children's who was in and out of treatment center, also was on olanzapine. A 15-year-old girl called in an immunologist. He ran the panel. The panel was positive. He treated her with plasmapheresis. In 2 weeks, she was back home playing tennis and off of olanzapine. We read the paper. We contacted him, asked for a sample to be retested, and all of her antibodies had returned to normal level. Unfortunately, I believe that this particular individual had a relapse afterwards. But again, as was talked about today, there are things to recognize and to watch, especially after patients begin to recover. So kind of in summary, if you think about this, as Dr. Fridd also showed, if you look at this autoimmune encephalopathy of infectious etiology, you can see PANDAS smack dab in the middle, autism, ADD, seizure disorders, and probably many others. And the other thing, if you remember this axis here, is that if you only treat the infectious agent or one of them, if they have more than one, you might see some improvement, but you may not see complete improvement. Or if they're treated for the immune system, but they aren't treated for the infectious triggers, you might see some improvement but relapse. So this is what makes it a bit more complicated than just treating what you see. And again, thanks to Tim Sorrell and his group, My Kid Is Not Crazy, and for bringing the DVD. I think if you pass that around and share that with as many people as possible, it's something that I think will be very, very enlightening. I'm going to leave you with this last thought. So things are not always easy. I show these two people, Maurice and Emile, and I don't know, I've not had anybody who actually knew much about them, but maybe, have you heard of them? Okay. Well, these two gentlemen were the engineer and the architect who were trying to propose a major structure for the 1889 World's Fair. And they're not famous, so to speak, but they had an idea, and they thought the idea of this structure would be a great idea to use for the World's Fair, but they couldn't convince their boss to actually build it. So they worked on him for about a year, and his name was Gustav Eiffel. And after about a year, he finally relented, but he did, I think the story goes that he indicated that he would name it after them and give them some royalties or something. I'm not sure how that story worked out, but I think that they had a challenge trying to get their boss to actually believe in it and do it. Well, as you know, it begins with a vision, continues with perseverance. And so the plans were drawn, and the plans were drawn. Once the plans were drawn, they actually began building. But they ran into problems because what they found was the structure in which they built it upon was not able to support a structure of that size because it had never been built before. So they had to bring in some problem solvers to fix the problem and figure out how to make the base and the foundation strong enough so that they could actually build this structure on top of it so it wouldn't fall apart. So they began building it, and then guess what? They ran out of money. And they ran out of money, and there wasn't a lot of people that were willing to actually help them with money. So they began going around trying to share their vision, explain all the things that they were going to do, how it's going to be a beautiful structure, all of that. And finally, they were able to actually raise enough money and build it. But the plans were to tear it down. In fact, they were getting ready to tear it down because it wasn't supposed to be a long-time structure. And in fact, people began to think it was a little ugly. They didn't like it. And so one thing that was interesting and serendipitous, and I think that's important to recognize because Louis Pasteur talked about that, and so did Alexander Fleming, who discovered penicillin, is that they took advantage of the World War. They found that if you put an antenna on the top of the Eiffel Tower, you can actually intercept and communicate farther than you could than anywhere else. And so that kept the structure from actually being torn down. And so what we see is one of the most recognized structures that we know today. And the point I want to leave you with is the idea began in the mind of an engineer and architect many years before anything was ever laid. And so with a vision and with perseverance, PANDAS, PANS, all these other autoimmune encephalopathies, I believe one day will be just treated as common and they'll be caught early. But it's because of the vision and the perseverance of you, PANDAS Network, the frontline physicians, the pioneers here that actually are doing the work. And so let me encourage you, because our mission is to help change how medicine is practiced for neuropsychiatric disorders. But as Winston Churchill said, never, never, never give up.